Here's a video of Mr. Phillips, and today we'll be looking at making recombinant DNA. Technology is obviously a up-and-coming area, and this topic, I'm just fixing my computer if it's banging around. This topic is a area obviously where it's probably out of date before I've even discussed it with you. But the idea is that many human diseases happen where individuals not being able to produce the correct proteins, for example. So traditionally we often inject these chemicals. If we can make pure copies of these proteins by taking a required gene out and insert it into a microorganism, this means we can have a better medical supply. So for example, this has been done for many years with insulin. Recombinant DNA basically means the DNA of two different organisms combined, and the organism we are producing is called transgenic. Basically, two reasons this works. One, because the genetic code is universal, so the same 3 letters in codons code for the same amino acids in all living organisms. And transcription and translation are the same in different organisms. So they're the same in a bacteria as they are in a human. This process will cover several videos. This is the first of the videos. The first stage of this process is isolation, which is what we're going to look at today. We'll then go into insertion. So how do you insert it into a vector, which in the A-level course is the plasmid? How do you get the plasmid into the suitable host cell, normally a bacterial cell in A-level? How do we identify whether the host cell has uptaken the gene, and then we obviously make it grow or. So the key thing, first of all, is we need to identify the gene we require, and we need to isolate the gene from the organism that we are going to want to make copies for, let's say, the protein, so insulin, for example. DNA is too long to be used completely, so we have to fragment it. And basically, there are 3 ways we isolate the DNA. We can use reverse transcriptase, which we'll talk about in a few moments, restriction endonucleases to cut DNA into small fragments, or the gene machine to artificially synthesize the DNA. So let's look at each of those in detail. Transcriptase, which turns messenger RNA into a single-stranded form of DNA. And you'll have heard about reverse transcriptase in your AS modules where you did HIV, and it's the enzyme that HIV uses to turn its messenger RNA into DNA, and then that incorporates with the host cells. So the reason we use this in several cases, and the most famous one obviously being insulin, is a very small polypeptide, about 51 amino acids in size, so it has less than 200 bases of DNA. This makes it incredibly hard to find the gene within your DNA. So we find it by looking at the beta cells, which will obviously be constantly using the gene, making messenger RNA in transcription, and then using that messenger RNA in translation to make the insulin protein. So we isolate the messenger RNA from the beta cells, and then we use the enzyme reverse transcriptase, which again is from retroviruses like HIV, and that reverses messenger RNA into a single-stranded copy of the DNA for that. We can use that and insert it into plasmids. The reverse transcriptase, let's have a look at the stages here. We start off with the beta cells, the islets of Langerhans. They make messenger RNA for insulin. We then take the messenger RNA coding for insulin. We act as a template to make a copied DNA strand, which if you get polymerase chain reaction later on in the next few videos, that will sort of explain how that can be done. We then take that single-stranded DNA and we DNA polymerase, and then we take that double-stranded DNA and we can insert that directly into the plasmid, which is on the next video. Type of isolation of the gene is using the restriction endonuclease, which is a type of enzyme to cut DNA into small fragments. And these enzymes have been isolated again from bacteria, which use them to defend themselves against viral infections, to stop the virus incorporating its DNA into the bacterial DNA. There are over 50 of them commonly used, and the key thing is they cut the DNA at specific base sequences, and it normally is less than 10 bases long how they do it. and it leaves something called a staggered cut. So here you can see we don't cut it down here and down here. It cuts the first strand of the DNA and then the second strand a few bases down, leaving these exposed unpaired bases on one end and on the other, which would obviously be complementary to each other, called sticky ends, and they'll become important. Some restriction enzymes do just cut straight down, but the ones that are most useful for us, such as EcoR1, which is the only one you probably ever hear of, cut in staggered cuts, leaving staggered sticky ends. And we call this a palindrome sequence. You see here AATT, and it's obviously the opposite TTAA on the complementary sticky end. And because DNA in all organisms has lots of similar codes, we could use the same restriction endonuclease to cut the desired gene as the plasmid to put it in, and therefore we can ensure that they will attach to each other. But that will come up. And the third stage is the gene machine, which is used to artificially synthesize DNA. So with the gene machine, we work backwards. We start with the primary structure of the protein. So we have our protein structures that we have identified, work out what the amino acid sequence is. From the amino acid sequence, we can then identify the base sequence of that gene, of all the codons in the messenger RNA to produce it. We then can program that sequence into a computer. We have to do ethical and biosafety checks. So ethical, for example, are we making something like a drug? Well, that might not be ethical. And biosafety, is this going to be a safe chemical compound we're going to use? The program then makes many small strands, which are really small, and they overlap, and then we have to stick them together. They're called oligonucleotides, which we assemble into a gene. This will not create any introns because obviously we're just using the amino acids, so introns are not taken into account, only exons, but that is fine because the bacteria doesn't have introns anyway, and it will just mean it will work. So with the gene machine, we then copy this gene once we've made it into lots of copies using polymerase chain reaction. See the upcoming video, and we'll get complementary strands added to it. Sticky ends will be then added, and it will be added into a bacterial plasmid to be cloned and make multiple copies in the bacteria. And the gene is then checked using standard sequencing techniques. The benefits of this is it takes a relatively short time frame. There's no introns, meaning the DNA can be transcribed by the bacteria. We don't have to have extra steps to effective. So again, let's have a look at the stages of it. You identify your primary structure of the protein. You identify the triplet sequence or codon sequence in the messenger RNA. You sequence it to a computer, do ethical and biosafety checks. You then make overlapping copies, such as here, which are called oligonucleotides. You assemble them into that gene has no introns, so it is put straight into polymerase chain reaction to make multiple copies, and then we insert them into the plasmid, and then that will be grown as we have talked about before. In the next video, we will continue to look at how we use these bits of gene that we've isolated in vivo cloning.